All right, so last time we talked about cellular respiration. Today we're talking about fermentation. The big takeaway from today's lecture is fermentation is about getting quick energy. It's about getting ATP very, very quickly. It's not very efficient, but it gets the work done. And also there's a little bit in here about secondary metabolites. Last time we talked about aerobic cellular respiration. So aerobic, what does it mean to be aerobic? With oxygen. It absolutely needs oxygen. It only happens if oxygen is present. I'm going to go through it very briefly again, the process. What's the first step? Step 1. Glycolysis. So with glycolysis, what molecule are we breaking down? That's right, we are breaking down glucose. We are breaking the bonds of glucose. If you take this 6-carbon glucose and you cut it in half, you have a 3-carbon molecule produced. What's that 3-carbon molecule called? Pyruvate. So you have 2 pyruvate produced. Net to a DP go in and to a TP come out. You also have 2 NAD plus go in and 2 NADH come out. So that's the whole of step 1, glycolysis. Step 2, the breakdown of pyruvate. So pyruvate's going to break down. It's going to use an enzyme called pyruvate decarboxylase to remove a carbon dioxide. Now we have 2 pyruvate molecules, so we cut 2 carbon dioxide off. And that releases 2 NADH. That pyruvate breaks down to an acetyl group. The acetyl group gets attached to coenzyme A to produce acetyl coenzyme A. So that's step 2, the breakdown of pyruvate. Step 3. That acetyl coenzyme A enters into what's known as the Krebs cycle, or the citric acid cycle. In this case, you're going to have this acetyl coenzyme A is going to bind with a 4-carbon molecule to produce a 6-carbon molecule, and then that's going to just be systematically cut apart. So you're going to release some carbon dioxide, and in the process, when you break that bond, you release some NADH. You're going to release some more carbon dioxide, and in the process, you're going to release more NADH. Cut off the functional group, and you get some GTP produced. Another, you get FADH2 produced. And another, you get some NADH produced. In this process, we've produced a whole bunch of energy intermediaries. We've produced a lot of NADH. Now, NADH is special because it has an excited electron. Does that make sense? The electron can do work. This Krebs cycle is happening in the inner membrane of the mitochondria. Now, this inner membrane has lots of twists and turns. What are these twists and turns used for? Increased surface area, so reactions can occur. Embedded in that inner membrane, you have these electron-accepting complexes. You're producing a whole bunch of energy intermediaries that then go to these complexes and drop off their electrons. They're like little dump trucks filled with electrons. They drive up, and they're like, here are these energetic electrons. Those electrons start to do work. What's the work those electrons do? They push hydrogen into the intermembrane space. And they jump from one molecule to the next in an electron transport chain and push more hydrogen across. And they push more hydrogen across. So what are we building up in this intermembrane space? An electrochemical gradient. The final electron acceptor, the reason we have to breathe is what? Oxygen. Oxygen accepts the electron, is converted into water. This only works if oxygen is present. Now, lots and lots of hydrogen is in that intermembrane space. Where does it want to go? It wants to head through ATP synthase to the center again. It wants to follow its gradient. So you use the power of that electrochemical gradient to spin the ATP synthase, converting ADP into ATP. It's extremely efficient. You produce lots and lots of ATP. You produce about 30 to 34 ATP at a time. An enormous amount per glucose molecule. Questions on that? That was last lecture. But there are going to be circumstances where you have no mitochondria. Circumstances where you have no oxygen. Can you guys think of a time where a cell would not have mitochondria, or a type of cell that has none? Prokaryotic cells, no mitochondria whatsoever. They have no membrane-bound compartments. They can't undergo aerobic cellular respiration. You might have a lack of oxygen. There are instances where we remove oxygen from systems. When we go to ferment things in big fermenting vats, the part of the goal is to remove the oxygen. If you've ever canned anything, you don't want oxygen present. Most of the time they get that little nice vacuum seal in there, right? They prevent oxygen from getting in, because what does oxygen do? I'll tell you. Oxygen, right? When you think oxygen, what chemical process do you think of? Oxidation. What's oxidation? It breaks down stuff. It steals electrons, right? So if you were to get oxygen in this canned material, that oxygen would break down those products in there. You don't want oxygen in there. You don't want oxidation to occur. So there are going to be instances where there's no oxygen. Heck, think about you guys. If you were to go swimming and you hold your breath and you swim as far down as you can, eventually you run out of usable oxygen. It's converted to carbon dioxide. Can you breathe? No, because there's no oxygen there for you. That's dissolved oxygen in the water, but you can't get to it. Did I tell you guys the story when I tried to breathe water? Yeah, don't do that. It's a bad idea. The solution to a lack of oxygen is a process known as fermentation. Fermentation is an anaerobic process. It happens without oxygen. It can happen in the presence of oxygen, but it can happen without oxygen. It does not rely on oxygen. It's simple and it's fast, and I think you guys will like the simple part of it. But it's still going to produce some ATP. And that's why I get it now, why I put the Roadrunner up. He is a very simple creature, yet also very fast, just like fermentation. You know, I've been trying to figure out why I put that bird on there for like 2 years now. It makes sense all of a sudden. So we're going to use this fermentation to convert glucose into a waste product and energy. And we can do it without oxygen. We can do it without organelles. This is the awesome part. It is a two-step process. And you already know one of the steps. Step 1, glycolysis. Step 2, waste product formation. I swear, whenever I say that, have you guys ever watched South Park? Do you know the underwear gnomes? Step 1, steal all the underwear. Step 2, step 3, profit. Okay, anyways. It's an old reference, and if you don't get it, you probably should. First step, glycolysis. 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 Breaking apart a carbohydrate. We're going to take a molecule of glucose and break it down into pyruvic acid or pyruvate. Those are the same thing. In order to get this process going, 2 ATP go in to start it. Sort of like kickstarts it. 2 ADP come out. The molecule that's produced, fructose diphosphate, is then split into 2 molecules. PGAL. That PGAL is going to then be broken down into pyruvate or pyruvic acid. In the process, NAD plus is converted to NADH, and 2 ADP go in and 2 ATP come out. Yes?
So, like, on the board where it says 2 ADP go in and 2 ATP come out, why is it like 2 ATP?
Ishmael?
Because you got 2 that you had before, and then, well, the 2 that got broken down, and then you got 2 2 new ones. So you only have 2.
Okay, so what he said, perfect. Well done. You totally knew that one. Well done there. This is your starting. You put in 2. Coming out, you've got 4. So your net total is 2. Plus 4 minus 2. All right, so we've broken down this glucose to make pyruvate. Questions on that? All right, you already knew that. Great. Now, that pyruvate, if it doesn't go into the Krebs cycle, if it doesn't get broken down through oxidative phosphorylation, it can be lots of different things. If it's exposed to the enzymes in Propionibacterium, it can become propionic acid. So Swiss cheese. Have you ever had Swiss cheese? You at least know what Swiss cheese looks like. It's in all the cartoons. Swiss cheese has all these holes in it. The way you get holes in Swiss cheese is you have this big block of cheese, right? These Propionibacterium are producing both propionic acid and carbon dioxide. So these holes are actually bubbles. When you cut through a bubble, it just looks like a circle. So Swiss cheese is built from, effectively, lots of these carbon dioxide bubbles. If it was to be introduced, this pyruvic acid was introduced to a lactobacillus, streptococcus, or bacillus, you get lactic acid. Now, in this course, you've looked at lactobacillus. Lactobacillus is in yogurt. This is what gives sort of that little bite that you get when you have yogurt. It's cheddar cheese. It's what makes cheddar cheese sharp. It's what makes soy sauce have that sort of fermented taste to it. It's lactic acid. Lactic acid. Oh, also super fun. You ever had sour gummy worms? That's where it comes from. Saccharomyces, yeast. If you were to introduce pyruvate to yeast, the enzymes in yeast, you produce carbon dioxide and ethanol. Wine, beer, bread, things like that. If you introduce pyruvic acid to Clostridium, the enzymes in Clostridium, you end up making acetone. Slight difference between acetone, which is nail polish remover, and ethanol. And if you introduce it to an Actobacter, you end up with acetic acid or vinegar. Any of these organisms can break down the same product, pyruvate, into lots of different waste products that can be useful or not. When you guys were younger, you've seen examples of this. You've seen lots of examples of this. Did you ever try to do you think to yourself, well, grape juice, if I let it sit out long enough, it's just going to become wine? Yeah, I think we've all done that at one point. It didn't work. What happened? It ended up turning to vinegar, really. What if you have wine? If you haven't had wine before because you're too young, you may have heard, what if you leave wine out for too long? What if you let wine doesn't stick around forever. What happens to it? You guys are pretty good at this game. You drink it. You drink it down. Actually, you try and drink it, and what you get is vinegar. You heard of red wine vinegar? White wine vinegar? They come about because that grape juice was introduced to this active. So we're going to look at 2 models for fermentation. We're going to look at yeast, and we're going to look at, I forget the other one. We're going to look at the lactic acid breakdown. Do you guys have any friends who might brew their own beer? They've tried. You guys have seen these like self-brewing kits? People try to do this, and it's hilarious to watch. There are a few things that always happen. Usually it blows up in their closet first off. But the big thing is, when these people come to you, when your friends come to you and say, I have just created a new microbrew, they're going to hand it to you, and they're going to say, drink this. You need to be very sure that your friend understands clean microbial technique. If you don't see them washing their hands often, they might have introduced something new. They wanted just yeast in there, but they may have introduced Clostridium. So instead of producing ethanol, they may have produced, acetone. And you don't want to drink that. It'll make you go blind. That was actually a big thing, when they talk about alcohol making people go blind. It's not because it's alcohol they're drinking. It's because the alcohol they produced was contaminated with another substance that broke it down into a different chemical altogether. the more you know. So there we go. First, one more thing. This is always, if you guys know any brewers, the way to mess with them. I do this not all the time, but sometimes. You go to a brewery and you say, "Wow, did you put licorice in this? Do I detect a hint of licorice or anise?" And the reason for that, or anise, I'm sorry. The reason for that is that's a byproduct of a different organism, and it means the process is flawed, so they have to clean everything. Anyways, just in case you wanted to really mess with somebody. Alcohol. Today's a practical lesson in biology. So alcohol fermentation. We're going to take yeast and convert it, use that yeast to convert glucose into ethanol. That's it. Glucose breaks down to ethanol. Two-step process. Now, we're going to use the waste products of this process more than we're actually going to use the energy from it as humans. Other organisms, yeast uses this fermentation to get most of its energy. For yeast, the whole point, the big point of this is to convert that pyruvate into a waste product so it can recover NAD+. I mean, that's literally all of fermentation. Fermentation right there. Glycolysis, waste product formation. That's it. For us, we can use it because that carbon dioxide that's released causes bread to rise. Have you guys ever baked bread before? If you bake bread, a lot of times you're going to bake the dough, pound the dough, fold it, do all these other things, and then set it down somewhere. Where do you set it? In general, have you guys ever had to sit bread that you don't just throw it in the oven, you let it rise. In order to let it rise, where do you put the bread in general? Good place. How would you make it rise faster? How about that? On a stove, somewhere warm. Why do you put it somewhere warm? Heat speeds things up. It speeds that kinetic energy, and the enzymes in the yeast are able to interact with the glucose, breaking it down, producing carbon dioxide. Faster, the warmer it is, the faster the temperature, right up until what? When does this suddenly, if you get too hot, the reaction rate decreases? When it denatures, when you go to temperature. Ethanol is released that we use for alcohol. Alcohol has actually been a major motivation for civilization. It doesn't just give us good ideas. Alcohol was very important for early civilization because alcohol was safer to drink than water. Alcohol kills a lot of microorganisms. So early civilization, you had a watered-down beer to drink instead of water because water was deadly. So what we need to do is we're going to take these 2 pyruvate, 2 ATP, and 2 NADH that were produced from glycolysis. Those 2 pyruvates are converted to acetaldehyde, just like over here. But instead of combining with acetyl-CoA, they're then broken down into ethanol and NAD+. That acetaldehyde becomes ethanol by these NADHs investing their electrons in it, getting that NAD+ reformed. So now you have a cycle. The whole point of fermentation is to give you some quick ATP and to recycle that NADH. Get it back to NAD+, so you don't have a rate-limiting step. So I want to emphasize that. All fermentation has a few things in common. And if this sounds like a good exam question, it's because it is. What do all forms of fermentation have in common? All fermentation is an anaerobic process. It does not require oxygen. All fermentation produces a waste product. And all fermentation recovers NAD+. It also produces some energy. So here's the big question. It's not the big question. It's a question. It's an application of this. We have this molecule of glucose. Now, in cellular respiration, we produce 30 to 34 ATP. We took the glucose and we cut it up into little bitty pieces. That's a lot of energy. In fermentation, we take this same molecule with the same amount of energy present, but in this case, we only are able to release 2 ATP. Where's the rest of the energy? Energy is neither created nor destroyed, so where is it? The rest of the energy is stored in the bonds of the waste product. Remember, you've got kinetic energy and potential energy, and you convert from one to the next. So the energy is stored in the bonds of the waste, which we can then go and recover through other processes. We can take that waste, break it down, ignite it, light it on fire, do whatever. We can get energy from it. So that's ethanol. You've also got lactic acid fermentation. Totally different process, but you'll see that they do the same thing. The point of this lactic acid fermentation is you take glucose and create pyruvate, glycolysis. You have your waste product formation where you make your lactate. In this case, you're converting NADH back to NAD+. It's all about recovering that NAD+. So vital to keeping this process going is to keep making that NAD+, because eventually, without, this one's backwards, sorry. Without that NAD+ being reformed, you can't have glycolysis happen anymore, which means you don't get any more energy. That's it. That's all fermentation. Glycolysis, waste product formation. The end. There are some organisms out there, though, that can switch between being anaerobic and having aerobic cellular respiration. Those organisms are known as facultative anaerobes. Yeast is one of them. Yeast it would prefer to have aerobic respiration. It wants to produce 30 to 34 ATP per molecule of glucose. But if there's no oxygen present, it switches gears and starts to produce ATP by burning glucose faster and having a lower amount of energy produced each time. You've got some cells that are facultative anaerobes. Your muscle cells are facultative anaerobes, which is really important. If your cells ran out of oxygen, if your cells had no oxygen, I don't know, you held your breath for too long, you still need them to work. You still need your heart to beat. So your cells suddenly switch from being aerobic to anaerobic, making as much energy as they can to keep your body going while you have no oxygen. That burning sensation you feel when you hold your breath too long, or if you've ever worked out. Has anybody ever worked out? Has anybody ever intended to work out? Okay. If you've ever worked out at any point, or, I don't know, you went up a flight of stairs and you were out of shape, you start feeling a burning sensation. Do you know what I'm talking about? So what that burning sensation is your muscles have used up all of their oxygen. So they've switched to an anaerobic process. So now they're burning through glucose faster and producing this pyruvic acid, in this case lactic acid, for our muscles. So you have this lactic acid buildup in your muscle cells. You start to feel this pain. How do you fix that pain? How does the pain go away? It takes time. It takes you breathing. Breathing can be difficult, I know, but you do it. So you've got your anaerobic bacteria, anaerobic organisms can switch, if they're facultative anaerobes, to using aerobic cellular respiration because it's more efficient. Questions? Now, surprisingly enough, not only is there aerobic cellular respiration, there's also anaerobic cellular respiration. Respiration that occurs without oxygen. Now, what's the whole point of oxygen and aerobic cellular respiration? What does oxygen do? It was over here that I just erased. It's converted into water, right? It's able to do that because it's the final electron acceptor of the electron transport chain. In anaerobic respiration, we can have chemiosmosis happen that movement of hydrogen ions without oxygen and without mitochondria. What's going to happen is you're going to produce this hydrogen gradient by pumping hydrogen out of the cell and then carrying the hydrogen back into the cell through ATP synthase. And you're doing that using nitrate as the final electron acceptor, which is reduced to nitrite. So here's how that works. I'm going to draw a picture. You've got a bacteria, right? No extra organelles in it. That bacteria, though, can have what's called an invagination. It's got an indention in it. It's got a little bulge going into it. Now, into this bulge, you've got a series of transmembrane proteins. You're going to use energy. You're going to use NADH that's made from the breakdown of glucose to push hydrogen across this barrier. And in the end, you're going to convert nitrate to nitrite. So far, so good? Doing the same, you're just taking these NADHs, these electron intermediaries that you produce from breaking down glucose, and you're using them, the electrons from those, to push hydrogen across this barrier. So what have we created here? An electrochemical gradient's building up. It's not in its own organelle or anything like that. It's just right there, outside the cell. But it's in a area. To get back into the cell, it goes through ATP synthase, converting ADP into ATP. Sort of all goes in like that. Does that make sense? You can do that without oxygen. So this is conducted entirely in the cell membrane, and it very likely evolved soon after we had real life on Earth. This was a use of the membrane in order to create energy to help the reactions occur for living organisms to survive. That hydrogen gradient is going to ultimately power ATP synthase. This is very, very common across the domains of life. It also provides a little bit of evidence for endosymbiosis theory. So if you've got these cool things, you can produce a whole bunch of ATP in that invagination, right? So you have another invagination. Now you're making twice as much ATP. You have another one, and you start to see a unique shape forming. Well, if this was eaten by a bigger cell, it forms vacuole around it. And eventually, you end up with a double membrane-bound organelle. This is likely how we got our mitochondria. So that's anaerobic respiration. Do you guys have questions? That's it. So that's cellular respiration and fermentation all wrapped up. That's your next 2 weeks of lab. Feel comfortable with it? In lab, you will need to draw flow charts. The flow charts we drew in class are the exact flow charts you need for your lab. If you don't have questions, I want to move on to a new concept called secondary metabolites. Secondary metabolites are one of my favorite things to talk about. These are going to be molecules that are produced for organisms, but not essential for development and growth. These are the extra things. These are the fun things. These are the things that give organisms an advantage, but aren't necessary. These secondary metabolites have been around for billions of years. As long as we've had life, we've had organisms that had to fight each other, organisms that needed an adaptive edge. Now, for us humans, we've got some cool adaptive edges. We can make tools. We make tools and we can beat things up and then eat those things. Plants can't do that. Certain animals can't do that. Bacteria can't do that. In order for bacteria and plants and other organisms to fight each other, they undergo chemical warfare. They need to, plants produce more secondary metabolites than any other organism on the planet. They need to defend themselves. Do you guys know what these are? Don't say flowers. They are flowers. What kind of flower? Poppies. Where do we know poppies from? What are poppies good for? Drugs. Sure. You can use poppies to make heroin. I was going to go with Wizard of Oz, but yeah, we can go with heroin. Same idea. Both actually are the same idea. Have you guys seen The Wizard of Oz? We'll also talk about heroin, don't worry. Yes, you've seen The Wizard of Oz. What happens when Dorothy goes running into a field of poppies? She falls asleep. What happens when you take heroin that you've seen on shows and stuff? You pass out, your body slows down. These plants are producing secondary metabolites to prevent predators from eating them, that slow down their heart rates. If an organism came and ate a bunch of poppies, their heart rate would begin to slow down. This tree, what do you notice about this tree? It's big. It's all by itself. It's all by itself, not because it's it's shading out other trees. The reason is these roots are systematically putting out poison to kill anything around it. You'll notice that a lot of time big trees are not surrounded by anything. They're all by themselves simply because they're killing everything around them. One type of secondary metabolite is called the phenolic. And phenolic compounds work by producing strong colors and strong flavors. The colors of daylilies, these are all phenolic compounds. Oh, you know what? Before I go any further, I just want to let you guys know. I will tell you the one question I will ask you about secondary metabolites. This is the one question. I just want you guys to sort of pay attention and find out, learn a bit about the world. Give 2 examples of secondary metabolites. I might say give 4 examples of secondary metabolites. That's it. Just give examples. Know what a secondary metabolite is. It's produced but not essential for growth and development. But I do want you guys, I don't want you to worry about taking 1,001 notes on this more. I want you to just see how the world works. Does that make sense? You got these bright colors. What are bright colors for in plants? Few things. One is attract bees, pollinators. The other is to say, don't eat me, I'm poisonous. They produce strong flavors. Those strong flavors are produced not so that the plant tastes better. That's actually the exact opposite reason they make these flavors. It's so the plants taste terrible. Cinnamon. Do you guys remember a few years ago the cinnamon challenge? No? Did anybody try to do that? What happens? Somebody died, that's true. You choke. You can't swallow it. This is a plant defense mechanism. Literally, the plant is producing these phenolics to stop you from eating them. So by taking concentrated plant phenolics, you are endangering yourself. Flavonoids are one of these big things. Chocolate and vanilla produce a lot of flavonoids. You guys ever had chocolate? And by chocolate, I don't mean the processed chocolate you get from the store, like a chocolate bar. I mean, you ever gone into your parents' pantry, found a thing of chocolate chips and thought, this will be delicious, I'll just eat this. And it turns out it was baker's chocolate? Yeah. Or a hot cocoa mix that hasn't been mixed yet with anything? Yeah, those chocolate tastes are these phenolic compounds, these flavonoids. Vanilla. Vanilla is so potent. It's there to stop organisms from eating it. Of course, we look at it and we're like, we're going to try it anyway because we rule the earth. These flavonoids play a major, major role in color. Another phenolic compound, the tannin, is responsible for the bitter taste of young red wines. Grapes. You guys have eaten grapes before? Red grapes? You're eating grapes right now. When you were younger, did you ever peel your grapes? Were you ever that kid? If you never peeled your grapes, for those people who are smiling because they definitely peeled their grapes, or even worse, had their parents do it, Those grapes that you're eating with no skin on them are little packets of sugar in that sugar water, and that's it. It's the skins that give them the flavors, that give them that bite. When you bite into a grape, the skin of a grape, it is bitter. And that bitterness is there to stop you from eating it. But they're also so delicious. If you were to take wine and press it, allow it to ferment, young red wines are very, very bitter because of these tannins. Another type, these tannins can be found in blackberries. There are two kinds of blackberries in the world, exactly two kinds of blackberries. Sweet, delicious blackberries and horribly bitter blackberries. And you can't tell the difference by looking at them. You've eaten blackberries before, right? And you reach in, you grab a blackberry, and you eat it, and it's bitter. What do? You spit it out, then what? Try another one, because you can't tell the difference. And then that next one's good, so what do? You try another one, and it's bitter, and then you're like, better have another. They produce those bitter flavors to stop organisms from eating them. Mice. Mice will eat blackberries like crazy when they're ripe. If they're not ripe, if they're not sweet and delicious, if they're not sugary and wonderful, they're very bitter. And what they end up doing is these tannins are poisonous. They will cause the mice to die. So mice avoid these things, and other organisms avoid them. Deer avoid them when they're not ripe. But once they're ripe, they eat them like crazy. Now, why would a plant have these same fruit be very bitter at one point in their life cycle and then very sweet at another? What's the point of that? Go for it.
When they're bitter, it stops animals from eating them, so it can mature,
Right.
where it can then reproduce, and it becomes sweet, so that animals then eat it to where the seeds can get.
That's exactly it. So at one point in their life cycle, the very bitter blackberries, they're going to be poisonous. They're going to be toxic. You don't want to eat them. They're going to hurt your for you, you're a big mammal. They'll just hurt your stomach, and you'll be like, oh, I shouldn't have eaten so many. That discourages organisms from eating those blackberries at that phase in its life. As they get more ripe, as the tannins break down and sugars increase, they become more sweet. It's also that these contain the seeds of the plant. The seeds have become mature. So what happens is you eat them, the seeds get passed through your digestive system, and when they do that, the acid in your stomach removes the seed coat so the seeds can start to grow. Then you go and conveniently excrete somewhere. Okay, you guys don't excrete wherever you feel like it, I guess. You don't sort of like, hold on, I got to go. Let me go in those bushes. But other organisms will leave behind their droppings filled with blackberry seeds that will then just start to grow in a nice little pile of fertilizer. Another type of phenolic compounds, the lignin. They give wood its strength. Then you got alkaloids. We're all done with phenolics, we're into alkaloids. Have you guys seen these things before? These are, they provide bitter flavors or poisonous toxins. Do you guys know what these things are? Have you seen that before? I'll turn off the light so you can see it better. What does that look like? Pardon? It's okay. You just mulch. It does look like mulch because mulch is a plant product. This is actually much smaller than mulch. It's tea. In case you've ever ripped open a tea bag, that's what it looks like. Tea provides bitter flavor and poisonous toxins. That's why we drink it, actually. You drink tea because it poisons you systematically. When do you drink tea? Does anybody drink tea? Yeah, okay. What kind of tea do you drink in general? Like, yeah, what type? Do you drink chamomile? Do you drink, I guess there's the generic. What does it do to you? How about that? When you drink tea? Yeah, what does it do to you? Does it increase your heart rate? Does it calm you down? Does it speed you up? Chills you out, right? You know why? Because it's acting in the exact same way heroin would act. It's literally, you're taking these watered down toxins that plants produce to slow down your heart rate to calm you down. I think that's pretty cool. We're just like, you know, this plant wants to kill me, but I'm just going to boil it. Drink its poison because that's how awesome I am as a human. And call it cool. Flip side is there are certain teas that speed you up. They're caffeinated, right? They wake you up. They get you going. Same idea. Other organisms, they'll get them going too fast. They're going to move on. They're going to die if their heart beating too quickly. Anyways, I just think it's such a human idea of, well, you're trying to kill me with this, so I'm just going to do it myself. Actually, it's not a human thing. It's an American thing. One type of alkaloid is capsaicin. This elicits a burning sensation to discourage mammalian predators. If you've had a pepper, a hot pepper, think jalapenos, habaneros, things like that, it hurts to eat, right? You take a bite out of it. It hurts because this capsaicin hits the receptors on your tongue, and all of a sudden, your eyes start watering. You don't want to eat any more of that plant. This is there to discourage predators from eating the from eating the peppers as they grow. Yes?
Does that mean it doesn't affect like reptiles or anything?
Or birds, surprisingly. Birds can eat peppers they don't have the receptors on their tongue. So mammals, they don't survive well with this. Reptiles don't care. Birds don't care. We've actually weaponized capsaicin because if plants can do it, we can do it. That's where you get pepper spray. That's where you get various crowd dispersal technologies. I have a capsaicin story. Let me tell you my capsaicin story. Any of you guys have a garden by chance? No? If you have a garden ever, you will understand this pain. I have a garden. My garden was filled with tomatoes because I like salad. No. My wife likes that I have to eat salad. So I have this tomato garden. I put it at the bottom of my hill 1 year, and then it was eaten by deer because deer eat everything. Well, I said, I'll outsmart the deer. So I bring all of the plants up to the on my deck. My deck is elevated 15 feet above the ground. Deer aren't an issue anymore. So I put my tomatoes in, on my deck. They're growing well. They grow really large. I go to my window. My bedroom window overlooks my deck, and I look out one morning, proud of my garden, and behold, in the garden is a squirrel. And the squirrel looks at me while holding a tomato. Now, this is a almost ripe tomato. Not really ripe, not ripe enough to eat yet, except apparently for the squirrel. The squirrel looks at me. I look at the squirrel. And I swear, we make eye contact this whole time. He goes, takes a bite, and then drops it. And then, still looking at me, gets another one. Takes a bite, and drops it. So I'm mad. I'm like, go get that squirrel. I open up the window and I yell at the squirrel. And the squirrel looks at me and he's like, you're not out here, whatever. So I'm fuming about this because I thought I was going to have some nice tomatoes, and they're just disappearing because squirrels keep eating them. It wasn't just that squirrel. There were several of his friends involved, too. And I look at my wife, who had just gotten up, and she sees this. She's like, what's wrong? And I explained to her about the squirrel, and she's like, oh, we should do something about that. And I was like, yeah, we should. And then I looked at her, and I say the words that always make her leave town. I said, I'm a biologist. I can fix this. And she said, look, just do whatever you're going to do. I'm going to take the kids and go to my parents' house. Just make sure the house is clean when I get home. I was like, oh, it'll be clean. It'll be clean. So she took the kids and she went out of town. And I enacted my plan. My plan was all about capsaicin. See, I knew capsaicin will impact mammalian predators. If a mammal comes along and takes a bite like a freaking squirrel, they're going to hate that. I didn't have any pepper out there. I only had tomatoes. So what I needed to do was either spend a lot of time in the lab and cross tomatoes with peppers, but I didn't want to do that. So instead, I went out. I was going to create a capsaicin spray to put on my tomatoes. And to do that, I had to go to the only place in town where you can pick up some good chemical reagents for producing chemical weapons, Walmart. So I went to Walmart. And I'm going down the spice aisle. I'm looking for anything I can that's got some kick to it. I've got red pepper. I've got cayenne pepper, chili powder. I'm throwing all these things in my cart because, God, this girl's going to get it. And I ran into a friend of mine there who was a chef, and I said, Chip, what's the hottest thing that they've got in the store? And he's looking at my cart, and he's like, oh, are you making some chili? And I was like, no, I'm going to take out the squirrel and let me explain the whole process. So 15 minutes after I ranted about the squirrel, he said, well, I guess there's some jalapenos up front, but I saw some ghost peppers and some habaneros as well. Those are really hot. I wouldn't recommend using more than one of them. So like, thanks, Chip, I really appreciate it. So I go to the front, and sure enough, there's a package of 6 ghost peppers and 4 habaneros. Well, they only came in packages of 4, so I was like, okay, let's just take them all. So I loaded up my cart, I go to checkout, and you know the people in the checkout line, when they talk to you, they sometimes ask, what you doing? And so I'm going to the checkout line, and the checkout person, who doesn't actually care, said, oh, are you making some chili? And I looked at them and I was like, yes. Yes, I am. That's exactly what I'm doing. I got my ingredients home, and I took them all and I put them into a blender. I took pepper and I threw it in. I took cayenne. I took ghost peppers. I was just like, yeah, I've got space in the blender. Stick all of them in. Add some water, for funsies. Turn the blender on. I've got this, what's called a slurry. It is just a big pile of brown mush. So that was good. I took that brown mush and I put it into a pot of boiling water. Because what boiling water will do, capsaicin is extremely stable at high temperatures. So by boiling that slurry, I was able to extract the capsaicin into the water. So it's not in the pepper anymore. So far, so good. Don't worry, I was safe. I put on safety glasses. Downside is apparently steam goes right around those things. So I'm crying. I mean, literally, I just tear-gassed myself, but I'm going to get the squirrel. I take it out of the, I've got this big thing of brown liquid bubbling. My house is toxic. It's making me cry. I take it, I put it into a centrifuge. You guys know what a centrifuge is? Centrifuge is something that spins around very quickly in order to separate out the materials. The heavy stuff goes to the bottom, like the peppers. The liquid that's light would go to the top. So I put it in the centrifuge that I have at home. And by centrifuge, I mean I put it in a 2-liter bottle, tied it to a rope, and swung it over my head as fast as I could. So I separate out this heavy, heavy pepper from the supernatant, from the liquid. And now I poured the liquid into a spray bottle, and I've got purified freaking capsaicin in the spray bottle. Went outside and I sprayed all the peppers. So far, so good. But then I thought, because you got to think like a squirrel, I have all this leftover pepper. What am I going to do with it? All that supernatant. So what I did with the pepper was I put it at the base of the plant, all around the bottom of the plant. Put it on the railing too, because what I thought was the squirrel is going to come over now, look at me in the eye, come over, grab a tomato. Nothing bad happening yet because he's just grabbing it, and take a bite. Well, that pepper will kick in, right? So he'll drop it and he'll go, oh, my tongue hurts, and start doing that cute little squirrel thing where they start going like that, except they just walked in the pepper too. So anyway, so I decided to do that. I was so proud of myself, I went to bed. I woke up in the morning, rubbed my eyes, realized there's still pepper on my hands, start crying, look out the window, and there are 3 dead squirrels. End of story. It's really potent stuff. Plants can mess you up. It's another one. Atropine. Atrine's another alkaloid. It's derived from nightshade, hemlock. You've probably heard of it, but it was also referred to in ancient Greece as belladonna. Women would take the extract of atropine, just like I extracted capsaicin from peppers. They would extract atropine from nightshade and put a drop in their eyes. And what that did was it dilated their eyes. It made their pupils huge. It was what's called doe-eyed, if you ever read Shakespeare. With just the right amount, they would have very large pupils. Too much, they'd die. The things people do for beauty, People look at me and they're like, so why would they even do that? I'm like, well, why do people wear high heels? Doesn't really make sense. Oh, fun story about atropine. A derivative of it is what the eye doctor that you go to, the optometrist, uses in order to dilate your eyes. So don't worry, if they get it wrong, you die. You've got all these different alkaloids. Caffeine, nicotine, morphine, opium. All of them are in the same category, by the way. Same class of chemicals. They're all used as a defense. These things speed you up or they slow you down. They make your heart beat super fast, so you get out of there. They slow you down so predators can catch you. They make you dull-witted. They do all of those things. We have these recreational drugs. And the point of those drugs is to alter your state of mind. For the plants, by altering the state of mind of their predators, they're able to get their predators to leave, to not eat them. Terpenoids are responsible for smells. Anything that goes into a gaseous state easily. The smells of flowers. A class of terpenoids, a carotenoid. Now, where have we seen a carotenoid? Carrots, right? They give fish those orange colors. They give plants, they're able to help plants absorb light. And the final class of secondary metabolites I want to talk about are the polyketides. Polyketides are highly toxic, very specific metabolites that inhibit protein synthesis. So if you have one bacteria living next to another bacteria, and they don't want to live next to each other for very long, one bacteria will secrete a polyketide. That polyketide will stop the ribosome from working. Why are ribosomes important?
Help make protein.
Right. If you stop the ribosome, suddenly you can't make any proteins. No proteins mean no enzymes. No enzymes mean no reactions. So bacteria can literally kill their neighbor by secreting these chemicals. Once we as humans found that out, we were just like, yeah, this is totally us. We created antibiotics. We found the way that bacteria and fungus attack each other, and we're like, we're in. We're going to take that, and we weaponized it, and now we have medicines that will only attack prokaryotic cells, that will only attack the ribosomes of bacteria. So when you take an antibiotic, your cells are fine. You've got the antidote. You're immune to it. But every bacteria in your body is not, and they get wiped out. Do you guys have questions? That is fermentation.